So now we're going to talk about the last two phases of cellular respiration. So the third stage is going to be called the Krebs cycle. It's also called the citric acid cycle. Either one of those is fine. So what's going to happen is if we look at our pictures here, here's our acetyl-CoA that was created from pyruvate oxidation. So this little section here is that pyruvate oxidation happening in the previous video. So what's going to happen is acetyl-CoA is going to get introduced into the citric acid cycle. And there's a lot of stuff going on in here, so let's look at each little part. So the first thing that you might notice is that we're creating carbon dioxide. Another thing that's happening is a bunch of electrons are getting harvested, right? We have three NAD plus getting converted into NADH. Then we are creating an ATP, and then even more electrons are getting converted, or harvested, I should say, over here using the FAD, okay? However, how many times is this going around? Hopefully you're thinking twice, because remember, 2 pyruvates were created, right? So 2 acetyl-CoAs were created. So a lot of carbon dioxide, tons of electrons are getting harvested, and 2 ATPs are being created, okay? Now, let's go to your notes and see if that makes sense. Acetyl-CoA gets introduced into the cycle, 2 more ATP molecules get extracted, and several electrons are going to get removed by reducing NAD plus to NADH, okay? So we have made ATP. We've only made 2 in this phase, but that's not really a lot, as you'll see in the electron transport chain. All right, so you're probably wondering, why do we keep doing all these NAD pluses to NADH? Well, what's going to happen is those are actually going to end up at the electron transport chain. So all those electrons are going to end up there in order to make the electron transport chain happen. Okay, so let's look at a picture of the electron. This is Daisy. She's scared of the camera usually, but apparently not now. Okay, so the electron transport chain, what's going to happen here is those electrons are going to end up here, right? There's our NADH, and what's going to happen is the electrons are going to jump from here to here to here to here, all the way down, okay? How are they going to get there? NADH has brought them there. And what's going to happen is it's going to drop off that H with the electrons, and then it's gone back to NAD+. Now, that NAD+ can be recycled and used in those earlier phases that we've talked about. Now let's talk about the electron transport chain. So what's going to happen is, as these electrons are going from one to the next, to the next, to the next, these big purple things are proteins in the membrane of the mitochondria. So this is that inner membrane of the mitochondria that's all kind of folded up. So what's going to happen is, as those electrons do that, they're going to create energy in those proteins, enough energy that's going to cause these H's to get pumped to one side of the membrane. So you can see they're doing that all over here, right? So what's going to happen as a result of that is we're going to get a concentration gradient. So we're going to have a bunch of H's on one side of the membrane and not a lot on the other. So what's going to happen is they're going to want to go from high to low. However, they're not going to be able to get through this membrane. The only way they can get through the membrane is by going through this thing that's called ATP synthase. As they go through that ATP synthase, they're going to cause it to turn. And as it turns, that's going to cause enough energy to be created to push together ADP and a P, and that's going to make ATP, right? So that's going to be how we make ATP through ATP synthase. This thing right here, this purple thing, is going to be that enzyme that we use to do that. Okay, a couple of other things that are happening here. Remember I talked way in the beginning about how the electrons are going to jump from one to the other, and they need to have an acceptor molecule. Well, here it is, oxygen, that we have breathed in. So the reason we breathe in oxygen is to accept those electrons, and it's going to combine with hydrogen as a result of this reaction, and that's going to create water as a byproduct as well. So let's look at your notes and see if that makes sense. Okay. So the electrons carried by the NADH transfer to a series of electron carriers that are going to take the energy from the electrons and use it to pump protons, remember protons are H+, across the membrane. And that's going to be used by ATP synthase to produce 32 to 34 ATPs. So all these other ones had 2, 0, 2 ATPs. Now we've made 32 to 34, so that's a lot, right? Okay, so how did that happen? So as those electrons get harvested, what's going to happen is they're going to get passed along that electron transport chain, and they're going to create energy that's going to cause protons to be released into that other side of the membrane. Okay, so what happens is those H pluses are going to want to come right back through, and they can go through that ATP synthase, and that's going to create ATP. The process is called chemiosmosis. So that's that whole turning process and pushing the ADP and P together to make ATP. Okay, so that's going to be cellular respiration. In the next video, we'll talk about anaerobic respiration. So that's what happens after glycolysis if there's no oxygen present.